Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 28th November 2020. As we are into the revision mode, we are revising chapter number 13, photosynthesis in higher plants. We have been reflecting upon what are the various factors that influence photosynthesis and how can we write the overall photosynthetic reaction in chemical form and the various pigments that influence photosynthesis. And in today's session we are going to talk about the processes that take place during photosynthesis. So the entire photosynthesis, as we have been discussing, would take place in two steps. One is light reaction or Hillman reaction or photochemical phase, and the second one is dark reaction or Blackman reaction, or what we say reduction stage. CO2 gets reduced to form starch. So let's see the first one, light reaction or Hillman reaction. And this is a reaction which includes light absorption. So compulsorily chlorophyll will be there, it absorbs the light, and when light is absorbed, the photolysis takes place, photolysis takes place. So the light splits. What it splits? It splits water. So second is water splitting. What happens when water splits? I have explained to you yesterday. H2O splits into H plus and OH minus ions. So assuming that just for our understanding I have taken 4 molecules of water, and all the 4 molecules of water will split into 4 H plus and 4 OH minus, and in the bargain 2 H2O, 2 molecules of water will be formed as byproducts, and oxygen is another byproduct. And then when we write OH minus, 4 minus we have written, that means 4 electrons are also formed. Along with these 4 electrons, H plus means proton, hydrogen losing an electron, having a proton. So we have got 4 protons and 4 electrons when the water splits. That means basically during light reaction, light gets absorbed by the chlorophyll pigments with the 400 to 700 nanometers range, and during this light absorption, water splits and forms the release of electrons and protons, and oxygen is the byproduct along with the water. So oxygen release, formation of high energy chemical intermediates. During this entire process of light reaction, ATP and NADPH will be formed. So the pigments are organized into 2 discrete LHC. What is LHC? Light harvesting complex within photosystem 1 and photosystem 2. LHC are made up of hundreds of pigment molecules containing all pigments except single chlorophyll molecule in each PS, that is photosystem. Going ahead, this is the one. So primary acceptor and the reaction center, that is PS1 or PS2, a lot of pigment molecules, and then photons. So the pigments in photosystem 1 and photosystem 2 absorbs the lights of different wavelength, as I said 400 to 700 nanometers. Single chlorophyll molecule makes a reaction center. In PS1, reaction center has highest peak at 700 nanometers, hence called P700. So PS1 is called P700, and PS2 reaction center has highest peak at 680 nanometers, so its PS2 is called P680. And the electrons that are released will have to follow a path, and the path in which the electrons move around making some changes is called electron transport system. So reaction center of photosystem 2, that is P680, absorbs light of 680 nanometers in red region, and causing electrons to become excited. So it is the PS2 which is the initiation. These electrons, as you are seeing here, so LHC, H2O split, H plus and 2H, and look at this. This is the electrons. So these electrons are accepted by electron acceptors, and then it enters into, these electrons enter into electron transport system and reach to LHC, light harvesting system 2. And once this electron acceptor sends the electrons to electron transport system, there is formation of energy, ATP. And from LHC, light harvesting control system, electrons enter into photosystem 1, that is P700. Again, the electron acceptor, having the electrons to flow, helps in conversion of NADP plus into NADPH, which is again energy rich molecule. So reaction center of photosystem 2 absorbs light of 680 nanometers in red region and causing electrons to become excited. These electrons are picked up by an electron acceptor which passes to electron transport system consisting of cytochromes. So basically cytochromes will play a huge role. Electrons are passed down the electron transport chain and then to pigment of PS1, as you are seeing. So they are entering into PS1. Electron in PS1 also get excited due to light wavelength of 700 nanometers and are transferred to another acceptor molecule having a greater redox potential, reduction and oxidation potential. When electron passes in downhill direction, energy is released. This is the downhill reaction, and ADP plus inorganic phosphate, ATP, is formed. This is used to reduce the ADP to ATP as well as NADP to NADPH, as you have observed. The whole scheme of transfer of electron is called Z scheme due to its shape. Look at the shape. Is it looking like a Z? Yes, it is a Z scheme. So starting from this, yes, Z scheme. So that is why it is called Z scheme, because of the shape. Photolysis. Photo means light, lysis means breaking. But light is not being broken, water is being broken with the help of light, photons. So water release electrons that provide electrons to PS2. Oxygen is also released during this process. So what happens when photolysis takes place? H2O, H plus, OH minus, and OH minus I explained to you how oxygen is formed, and 4 electrons are released. So if you see here, there are 2 phosphorylations. One is the cyclic photophosphorylation, another is the non-cyclic photophosphorylation. So what is the difference between cyclic and non-cyclic? Cyclic photophosphorylation, it is performed by photosystem 1. Look at this. It's starting here, okay, and going to electron acceptor, taking a turn, electron transport system, reaching back to P700. So this is a circle. Hence we say cyclic photophosphorylation. It is performed by photosystem 1 independently. What about non-cyclic? Z shape, just now we said. It is performed by collaboration of both PS1 and PS2, photosystem 1 and photosystem 2. And this is not cyclic because it is in the Z shape. Second point, under cyclic photophosphorylation, an external source of electron is not required. There is no requirement of external source. Here the process requires an external electron donor. So we require a donor, electron donor, external electron donor. It synthesizes only ATP in photosystem 1, whereas here it synthesizes ATP as well as NADPH. So both are there in non-cyclic photophosphorylation. Under cyclic photophosphorylation, it occurs only in stroma or intergranal thylakoids. Remember grana? intergranal, between the grana. It occurs in the granal thylakoids, only in grana. No stroma is involved. Next, we go ahead with the chemismatic hypothesis of ATP formation. This hypothesis was proposed by Mitchell in 1961. ATP synthesis is linked to development of proton gradient across the membrane of thylakoid and mitochondria. So, proton gradient will decide the ATP synthesis. The process that causes development of proton gradient across the membrane is, look at this here. What makes the proton gradient? See, H plus, H plus, H plus. So, this is the lumen, and this is the thylakoid membrane. The pink one is the thylakoid membrane, and the stroma is outside, and the ADP, with the help of ATP synthase, the F1, F0, F1 particle in the grana. Okay, remember, in the grana, the ATP is synthesized. So, for this to happen, photosystem 2 as well as photosystem 1 are involved with cytochrome B and cytochrome F. And of course, in the process, not only ATP is synthesized, even NADPH is synthesized. So, what is required for this proton gradient to build up? Splitting of water molecules occurs inside the thylakoid to produce hydrogen ion or proton. So, first criteria is water should be split, then lot of H plus. As electron passes through the photosystems, protons are transported across the membrane because primary acceptor of electron is located towards the outer side of the membrane. Look at this primary acceptors. And third, the NADP reductase enzyme is located in the stroma. Stroma is outside, okay? Stroma side of the membrane. Electrons come out from the acceptor of electron of PS1. Protons are necessary for reduction of NADP plus to NADPH. So, these protons are also removed from the stroma. This creates proton gradient. So, removal of H plus both through the membrane as well as through NADP creates a lot of proton gradient across the thylakoid membrane along with the pH in the lumen. So, the gradient is broken down due to movement of proton across the membrane to the stroma through transmembrane channel of F0, that is F0 F1 we said, F1 particle we said, no, F0 of ATPase. One part of this enzyme is embedded in membrane to form transmembrane channel. You can see the embedment of one part, F0 is embedded and F1 is outside. The other portion is called F1 that protrudes, as you are seeing here, that protrudes onto the outer surface of thylakoid membrane, which makes the energy packed ATP. So, this is the one F1 particle making the production of ATP. So, ATP and NADPH produced due to movement of electrons is used immediately to fix CO2 to form starch or sugar. So, the product of light reaction used to drive the process leading to synthesis of sugar are called biosynthetic phase of photosynthesis. So, moving ahead, we have got what is called Kelvin cycle or C3 cycle or reproductive pentose sugar phosphate pathway. So, we are thankful to these two scientists, Melvin, Kelvin and Benson, and their colleagues used radioactive 14C. Normally carbon is 12, no? 12 carbon. So, 14 carbon and chlorella and syndesmus algae to discover that first CO2 fixation product is 3 carbon organic compound, that is 3-PGA, 3-phosphoglyceric acid. PGA stands for phosphoglyceric acid. Later on, a new compound was discovered which contained 4 carbon called oxaloacetic acid, that is AAO, oxaloacetic acid. In fact, we can write it as OAA, oxaloacetic acid. On the basis of number of carbon atoms in first stable product, they are named as C3 pathway and C4 pathway. Kelvin cycle can be described under 3 stages: carboxylation, reduction and regeneration. So basically these are going to help the CO2 to get fixed into starch. So basically it is reduction division. So I think in our next session we will be talking about this in detail. Until then,